So, till now we have studied how we can make the recombinant DNA molecule and insert it into the DNA. That means basic steps of the DNA technology. First is the selection of the gene of insert, then put it in the cloning vector, then transformation into the host cell, then selection, and finally the multiplication. The first recombinant DNA molecule was produced by different scientists named as Polberg, Herbert Boyer, and Annie Chang with Stanley Cohen. In year 1973. The first recombinant DNA molecule was produced by Polberg, Herbert Boyer, Annie Chang, and Stanley Cohen. And now we will move to the tools of recombinant DNA technology. The major tools of recombinant DNA technology are: first, the various enzymes involved. Second is the vector molecule, and third is the host cell, as we have discussed in the steps of the recombinant DNA technology. And the foremost important are the enzymes. As we know that enzymes are very specific in their function, and the most important enzyme of recombinant DNA technology is restriction endonuclease. Very, very important for recombinant DNA technology. What is this restriction endonuclease, and where it is present, and what is its function? Actually, restriction endonuclease are the enzymes. These restriction endonuclease, which are the basic enzymes for the recombinant technology, are present in many bacteria, are found in many bacteria, and in few of the fungi also. Why they are present in the bacteria? Actually, these restriction endonucleases serve as the defense system for the bacteria. Now the question arises, how they work as a defense system for the bacteria, and then how they can be used in the RDT. So, first we will study about the restriction endonucleases. Restriction endonucleases are very specific in their function as most of the enzymes. Actually, these are those particular enzymes, as the name indicates. Endonuclease means inside, nuclease cleaving the DNA or RNA, and here the case is DNA cleaving DNA. Means when we have a particular DNA segment, these restriction endonuclease can cleave in this DNA, but whether it is random cleavage or it is a specific cleavage. Yes, it is a specific cleavage. Even we say it is a very, very specific cleavage. And how this specific cleavage is done, how this particular restriction endonuclease cleave at a particular site, this is their recognition sequences. in a large nucleotide sequence, a particular restriction endonuclease can selectively identify the particular nitrogenous base sequence, and that is known as recognition sequences. If that particular recognition sequence, for example, a particular restriction endonuclease cleaves at GAATTC from 5' to 3' end, if this particular A is replaced by any other, now that particular restriction endonuclease cannot cleave this particular recognition sequences. So they are naturally present in the bacteria for their defense system. And how does this act? Actually, restriction endonuclease works under the two processes. One is known as restriction and another is known as modification. Say this is a bacteria which is having its own genomic DNA, and these are the restriction endonucleases which is present in this particular bacteria. Whenever any foreign agent attacks this particular bacteria, what is this mode of action? This bacteriophage inserts its genome into the bacteria. This is the genome of the virus. And whenever these restriction endonucleases which are present in it, they recognize their sequence, they get attached to those sequences and thus cleaves these foreign DNA. Fragmented DNA of phage. And in this way, this particular restriction endonucleases protect this particular bacteria from the attack of this virus, and thus it restricts, meaning is it restricts the entry of foreign pathogen. So what is the difference? Now the question arises. This is also the DNA sequence, and why this particular restriction endonucleases, which is very specific in their function, could not recognize the sequence which is present in this host DNA? Actually, what happens if this bacteria have to protect itself from its own restriction endonuclease, it undergoes the system of modification. What happens? The host bacteria undergoes methylation. And what does that mean? Undergoes addition of this group. And whenever this modification occurs in this particular DNA of the host cell, methylation occurs at various sequences. And now the sequence which is to be recognized by this restriction endonuclease now cannot be properly recognized, and therefore It protects its own DNA from the attack of restriction endonuclease, and it is known as restriction modification system. So, next is how does this recognizes the sequence, and what are the recognition sequence? Actually, the recognition sequences are generally 4 to 8 base pairs long palindromic sequences. Now, what is a palindromic sequences? Palindromic sequences means they read forward and reverse same. For example, Malayam, Malayalam means from this side and from this side they are exactly same. And when we said about the recognition sequences of restriction endonucleases, they should be same in 5' to 3' direction, and similarly in the anti-parallel strand 5' to 3'. So, this is the recognition sequence of the restriction endonucleases. Now, the nomenclature and the restriction endonucleases recognition sequences. The first is the nomenclature. This is important. How a particular restriction endonuclease got its name. For example, there is a restriction endonuclease which we got from Escherichia coli bacteria. So, that particular restriction endonuclease which we got from E. coli, how it is named? The first example of restriction endonuclease from this is ECO R1. E means the genus, CO, that are the species, Escherichia coli. R is the strain. We have already studied that a particular bacteria can have different strains. For example, in the Griffiths experiment, we have studied that Diplococcus pneumonia have two strains. One is rough strain and one is smooth strain. And what is this one? This is the sequence of the restriction endonuclease number obtained. Means how many restriction endonucleases we have already obtained from this particular bacteria. If it is one, this means this is the first restriction endonuclease which we got from this particular bacteria. So this is all about the nomenclature of the restriction endonuclease. Now we will discuss the recognition sequences. We have a large number of the bacteria from which we isolate the restriction endonucleases. For example, E. coli, Haemophilus influenza, then it is Providencia stuarti, Streptomyces albus. These are the different bacteria from we get different restriction endonucleases from this. For example, we got E. coli, Haemophilus influenza, Hen2 and Hen3. From PSA, from Streptococcus albus, we got SAL, and now these particular restriction endonucleases are having their different recognition sequences. So we will now discuss about the recognition sequences. Recognition sequence of EcoR1 is GAATTC 5' to 3', and in the reverse direction it is 5' to 3'. As G always pairs with C, A always pairs with T. Similarly, this is the recognition sequence of EcoR1. Whenever this sequence is present in the DNA, this can be cleaved by EcoR1. And what is about ALU1? It is AGCT. This is 4 nitrogenous basis long and this is 6 nitrogenous basis long. Whenever this EcoR1 detects this particular sequence, it cleaves it in this manner. Means it from 5' end it cleaves after G. Similarly here it cleaves after G. While in ALU1 it cleaves after this, and here it cleaves after this. Similarly, we have on the basis of these two different cleavages, the resultants are different. The sequence which is recognized by EcoR1 is GATTC, and while this is recognized by ALU1. ALU1 cleaves at this sequence, but the resultants are different. So this is DNA sequence. The resultants are different. How? 5' G, and in this is C, T, T, A, and here it is C, T, T, A. Here it is G. And by the cleavage of ALU1, it is AGCT, and here it is and these are known as different ends. This is staggered cut and this is even cut. So firstly we will now discuss about the detail of staggered and even cuts. Cleavages. So when a particular DNA sequence is cleaved by any of the restriction endonucleases, it can give two type of the results. Whether it will result into the staggered cut, that is also known as uneven, protruding ends, sticky cuts, etc. Means one is hanging over and another is known as blunt ends, symmetrical ends, this is also known as cohesive, means sticky, even ends. So with the cleavages which we have shown in the previous figure, EcoR1 or Hind3 gives these types of the cut, while ALU1 give this type of the cuts. Now the question is difference between staggered cuts and the blunt ends. So with that diagram we can clearly see when a staggered end is made, the resultants are as these. Means one end of DNA is overhanging over the other end, while when it is cut as a blunt end, both ends are equal. This is 5 prime, this is 3 prime, this is 5 prime, this is 3 prime. And at 5 prime end we always have a phosphate group with us, while at 3 prime end we have hydroxyl group with us. Similarly here we have hydroxyl group and here we have the phosphate group. While when it is blunt end, similarly this is 5 prime, 3 prime and this is phosphate group is here, hydroxyl group is here, hydroxyl group is here and phosphate group is here. And these ends which are extended ones are known as overhangs, which help in cohesion. That means it can easily convert itself into this circular DNA by joining as such. Say it is also this can complementary form, means A can bind to this T, this A can bind to this T can bind to this A, this T can bind to this A and this C can bind to this G. So this form like a structure and they can easily recoil, while this cannot happen in base Therefore, it is known as sticky ends. We have its own benefits and disadvantages. Means it can easily help us in the formation of recombinant DNA, as it have the complementary ends. But the problem also arises that every time it can self-coil itself. So this is all about the sticky ends and the blunt ends. Now this is for today, and we will move with the next topic in the next lesson.